Welcome to G Fun Facts Online. Today we are diving deep into a topic that sounds like it was ripped straight from the pages of a science fiction novel. But I assure you, it is happening right now inside the human body. We are talking about the brain, about epilepsy, and about a new biological protagonist that has emerged from the shadows of neurobiology to challenge everything we thought we knew about neurological disease. We are talking about zombie cells. Now before you roll your eyes, I am not talking about Hollywood monsters. I am talking about a very specific, terrifying cellular state known as senescence. This is the story of the silent insurrection occurring in the brains of nearly 1/3 of epilepsy patients, those suffering from drug-resistant epilepsy. Let's set the stage. The human brain is an electrified city. Order is maintained by a delicate, millisecond by millisecond balance of excitation and inhibition. It is a choir where neurons fire in synchronous harmony to process your thoughts, your memories, and your movements. But in epilepsy, this harmony is shattered. For decades, scientists have hunted for the root cause of this shattering. They blamed genetic mutations. They blamed misfolded ion channels, but recent breakthroughs in 2024 and 2025 have illuminated a different culprit. The culprit is the glial cell, the support staff of the brain, that has entered a state of suspended animation. These cells refuse to divide, yet they refuse to die. They linger in the neural tissue, spewing a toxic cocktail of inflammatory chemicals that poison their neighbors and rewire the architecture of thought. Today we are going to dissect the anatomy of these cellular zombies, understand how they create what we now call senescent synapses, and look at the revolutionary therapies that might finally cure the incurable. To understand the solution, we must first understand the anatomy of the undead. What exactly is a senescent cell? Cellular senescence is actually an evolutionary mechanism designed to keep you safe. It is a double-edged sword. When a cell accumulates too much DNA damage or stress, it pulls an emergency brake. It enters a permanent state of cell cycle arrest. It stops dividing to prevent itself from turning cancerous. In the rest of your body, your immune system typically spots these dormant, damaged cells and eliminates them. It takes out the trash. But in the brain, and particularly in the epileptic brain, this clearance system fails. These cells accumulate, and they undergo a monstrous transformation. The defining feature of a zombie cell is not its silence, it is its noise. These cells develop a hyperactive secretory profile, known as the SASP, or the senescence-associated secretory phenotype. I want you to imagine a factory that has stopped producing useful goods, like cars or phones, but continues to pump thick, black, toxic smoke out of its chimneys, day and night. That is a senescent glial cell. It pumps out pro-inflammatory cytokines like IL-1 beta and TNF-alpha, which inflame the surrounding tissue. It releases chemokines, which are signals that recruit immune cells, often causing collateral damage to the brain structure. It even releases proteases, enzymes that chew through the extracellular matrix, effectively dissolving the physical glue that holds neural networks together. But it gets worse. These zombie cells are metabolically deranged. They shift away from efficient energy production. Instead of using oxidative phosphorylation, they switch to glycolysis. They become sugar hogs. They consume vast amounts of glucose while producing very little energy. This creates a metabolic sinkhole in the epileptic focus. They essentially starve the nearby healthy neurons of the fuel they need to maintain their electrical stability, making those healthy neurons unstable and prone to misfiring. You might ask, why don't they just die? Why doesn't the body kill them? The answer is that senescent cells upregulate pro-survival pathways, specifically the BCL2 family of proteins. These pathways make them impervious to apoptosis, or programmed cell death. They build a biological shield around themselves. This resilience is what makes them undead, and it explains why the inflammation in epilepsy is chronic and unrelenting. Now, let's talk about the specific actors in this tragedy. In the context of epilepsy, the most dangerous zombies are not the neurons themselves. Neurons rarely undergo this type of senescence. The villains are the glia, the astrocytes, and the microglia. Let's look at the zombie astrocyte first. Astrocytes are usually the heroes of the brain. They wrap around synapses in what we call the tripartite synapse. Their job is to vacuum up excess glutamate, which is the brain's main excitatory neurotransmitter. If you leave too much glutamate in the synapse, the neuron gets overexcited. The astrocyte prevents this. But when an astrocyte turns senescent, often triggered by the stress of initial seizures, it fails as a gatekeeper. It downregulates the transporter pumps responsible for clearing glutamate. The result is a synapse flooding with excitatory fuel. A spark becomes a wildfire because the firefighter, the astrocyte, has walked off the job. Furthermore, these zombie astrocytes lose the ability to buffer potassium ions. High extracellular potassium depolarizes neurons, bringing them closer to the firing threshold. It is a classic trigger for seizures. They even retract their end feet from blood vessels, causing the blood-brain barrier to become leaky, allowing toxins from the blood to invade the brain. Then we have the zombie microglia. If astrocytes are the firefighters, microglia are the gardeners. They prune weak synapses to keep circuits efficient. But when they become senescent, they become confused and aggressive. They overexpress complement proteins that tag healthy synapses for destruction. They engage in something called synaptic stripping. But instead of pruning weak connections, zombie microglia begin to strip away inhibitory synapses. These are the brakes of the brain. When you remove the brakes from a neural circuit, you get runaway excitation. You get a seizure. This leads us to the concept of the senescent synapse. This term describes the functional ruin left in the wake of zombie glia. It is a synapse that is structurally present but functionally corrupted. It is the epicenter of drug resistance. Recent research has shown us 3 distinct failures at these synapses. First, there is a presynaptic failure known as snareopathies. For a neuron to fire, it must release vesicles of neurotransmitter using a complex of proteins called snares. In the toxic soup of the zombie cell's secretions, these proteins get oxidized by nitric oxide and reactive oxygen species. This leads to a leaky release of glutamate. Imagine a dripping faucet of excitation that keeps the postsynaptic neuron constantly on edge, ready to snap. Second, we see a postsynaptic deficit. Paradoxically, while the synapse is flooded with glutamate, the specific receptors that mediate learning and memory, the NMDA receptors, often shut down or become hypoactive due to the chronic stress. This creates a dual pathology. You have hyperexcitability, driven by dumb receptors that just shout fire, leading to seizures. But you also have cognitive decline, or brain fog, because the sophisticated NMDA receptors required for forming new memories are offline. This explains why so many epilepsy patients suffer from severe memory issues. Their synapses are stuck in seizure mode and cannot enter learning mode. Third, there is structural erosion. The physical scaffold of the synapse relies on cytoskeletal proteins. The inflammatory SASP degrades these structural beams. The synapse becomes wobbly. It forms transient, unstable connections that are prone to synchronizing inappropriately, which is the electrical signature of epilepsy. Now, here is the billion-dollar question. Why do standard drugs fail? Why is this condition called drug-resistant epilepsy? The existence of senescent cells provides the answer. Most anti-seizure medications target voltage-gated sodium channels or GABA receptors. However, the inflammation from zombie cells can actually alter the expression of these channels. If the lock changes shape, the key, your medication, no longer fits. For example, senescent inflammation can cause neurons to swap out drug-sensitive GABA receptors for drug-resistant variants. Furthermore, zombie cells engage in transporter overexpression. In their survival mode, they often upregulate P-glycoprotein transporters. These are biological pumps that eject toxins from the cell. In the epileptic brain, these pumps are often found on the blood-brain barrier capillaries, influenced by those senescent astrocytes we talked about. They actively pump anti-seizure drugs out of the brain before they can even reach their target. And finally, there is the network effect. Drugs act on individual receptors, but senescence alters the network topology. When zombie microglia strip the inhibitory brakes, the network rewires into a hypersynchronous state. No amount of sodium channel blocking can stop a network that is structurally wired to explode. There is a fascinating parallel here from the world of entomology that really drives this point home. There is a fungus called Entomophthora muscae that infects fruit flies, turning them into zombies. The fungus invades their nervous system and hijacks their locomotor pathways. It forces the fly to climb to a high point, a behavior called summiting, before killing it to spread spores. While epilepsy is not a fungal infection, the mechanism of hijacking is eerily similar. Senescent cells hijack the brain's plasticity mechanisms. Just as the fungus forces the fly to climb, senescent glia force the brain to seize. They co-opt the mechanisms of synaptic scaling and homeostasis, twisting them until the only output is a hypersynchronous discharge. The zombie moniker is not just a cute nickname. It reflects a parasitic relationship, where a subpopulation of damaged cells dictates the fate of the entire organism. So, if the problem is zombie cells, the solution seems simple, right? Kill the zombies. This is the premise of senotherapy, the most exciting frontier in epilepsy research today. Unlike current drugs, which only suppress symptoms, senotherapies aim to modify the disease by eliminating the root cause. The primary weapon in this new arsenal is senolytics. These are drugs that selectively induce apoptosis, or death, in senescent cells while leaving healthy cells unharmed. They do this by temporarily disabling that BCL2 pro-survival shield we mentioned earlier. The gold standard combination right now is dasatinib plus quercetin, or D plus Q. Dasatinib is a leukemia drug, and quercetin is a plant flavonoid found in onions and apples. In landmark studies published in late 2025, treating epileptic mice with D plus Q cleared 50% of senescent glia. The result was a dramatic reduction in seizure frequency, improved memory, and in some cases, the complete cessation of epilepsy. But we don't always have to kill them. Sometimes we can just muzzle them. This is where senomorphics come in. These are drugs that don't kill the cell but block the SASP. They stop the factory from pumping out the black smoke. Drugs like rapamycin, which inhibit the mTOR pathway, can suppress the inflammatory output of senescent glia, restoring synaptic stability without removing the cells themselves. The beauty of senolytics is the potential for a hit-and-run strategy. Because senescent cells take weeks or months to accumulate, you wouldn't need to take a pill every day. A patient might take a course of senolytics for 3 days to clear the trash, and then remain drug-free for months until the burden reaccumulates. This would drastically reduce the side effects associated with chronic daily medication. So are we ready for human trials? We are closer than ever. Analysis of brain tissue removed from patients with temporal lobe epilepsy has confirmed a massive burden of senescent astrocytes and microglia, up to 5 times higher than in healthy brains. The race is now on to develop non-invasive biomarkers, like PET scans that light up senescent cells, or blood tests that detect specific SASP factors leaking from the brain. The vision for 2030 is incredible. Imagine a patient enters the clinic after their first seizure. Instead of just being put on carbamazepine and sent home, they undergo a senescent scan. If the scan lights up, they receive a short course of targeted senolytics to nip the epileptogenesis in the bud, preventing the rewiring of the brain before it becomes permanent. For too long, we have treated epilepsy by trying to silence the shouting neurons, ignoring the toxic environment that makes them shout. The discovery of senescent synapses and the role of zombie glia changes the paradigm. It shifts our focus from the victim, the neuron, to the instigator, the glial environment. By targeting these undead cells, we are not just suppressing seizures, we are restoring the ecosystem of the brain. We are clearing the smoke, rebuilding the scaffold, and giving the synapse a chance to heal. The era of fighting zombies in the brain has begun, and with it, the hope that drug-resistant epilepsy may finally meet its match. That is it for today's Deep Dive. Thanks for listening to G Fun Facts Online.